Hi, everyone. My name is Carolina Makowski. I am an assistant professor in the Department of Psychiatry at the University of California, San Diego. I have been within the ABCD consortium since 2017, so it's so amazing to be at this point now with all the data that has been collected. And I'm really excited to contribute to the ReproNim course with an extended conversation around responsible use of data and statistical considerations with this data set. So let's dive in. So here are some objectives for today, and we really will be focusing on brain behavior associations, how we can interpret effect sizes, and differentiating between different methods that we can use to assess brain behavior associations. So for instance, with univariate versus multivariate methods. I also hope by the end of this session that you'll be able to really critically assess the impact of different demographic covariates and subsampling strategies on brain behavior results, as well as start to kind of create your own study designs that leverage ABCD study data and integrate them with other data sets. So this has, I hope, likely been highlighted already in previous ReproNim lectures, but effect sizes in ABCD tend to be very small. So just to kind of give a refresher on effect sizes, you're really measuring the strength of the relationship between two variables of interest. This is a kind of seminal way of defining effect size strength from Cohen. So typically people will report things like R, right, or R squared when you have two quantitative variables. And depending on what R squared value you get, you can categorize that into small, medium, large effects. Same thing with Cohen's d if you have, group differences that you're testing with a t-test, etc. And prior to ABCD, there had been large studies, right? But the ABCD is such an unprecedented sample size. So we're really starting to characterize true effect sizes that we might actually approximate in the real population. So they have been much smaller than I think what people had thought effect sizes would be with ABCD. So I've just highlighted a couple papers here from folks within the ABCD study consortium, Wes Thompson, Hugh Garavan's group. And just highlighting here, even from Hugh Garavan's paper in PLOS ONE in 2021, they had actually looked at hundreds of variables just within baseline ABCD data. No imaging here, but variables that are coming from like physical health, mental health, cognition. And if you carried out Pearson's correlations on hundreds of variables, the median in-sample effect size was just 0.03. So if you port that over into the size or the strength of that effect size, this would be very, very small, right? But it does not mean that these effect sizes aren't meaningful. It just means that you do need to have proper methods to detect them. And also, there are considerations around reproducibility of such small effects. So this is very similar when you incorporate the brain. When you measure associations between brain and different behavioral variables, effect sizes are very, very small. And folks that are working in neuroscience like myself, it has really been a huge goal of ours to understand, how the brain maps onto complex traits in humans to help us understand the brain and also the behavior in both typical and atypical development. So non-invasive magnetic resonance imaging has really been helpful in expediting efforts to understand these links between, for instance, neurocognition and psychopathology and the brain, because you can get these large sample sizes like we do with ABCD without actually going in and doing surgery or anything, right? And the costs are also coming down with MRI. But following what we have seen in other fields, for instance, in psychology as well as in genetics, it seems like we are facing this replication crisis in MRI or brain MRI behavior associations. And this could be due to many factors, such as huge variability in imaging measures and how people are running their pipelines and decision points they're making in terms of how they're actually acquiring imaging data. Variability in the behavioral measures, I really want to highlight that because for a while when we did see, headlines come up around these concerns with imaging data. MRI was the one to blame, but there's also a large variability in how we're collecting behavioral data, right? Sample size is a really, really critical point to consider when we're thinking of effect sizes, as well as I alluded to, which sorts of methods we're actually using depending on our question of interest. And kind of bringing back those challenges to the forefront and really identifying where those came from. As I'd already mentioned, brain behavior associations are very small, small in ABCD. There has been, a lack of reproducibility of these effects for a long time, but these concerns sort of resurfaced with a very high impact paper in 2022 that was published in Nature by Scott Marek and Brendan Turvo-Clemens from Nico Dozenbach's group. And their paper really made headlines around the world and suggested that you required thousands of individuals for brain-wide association studies. And there were other studies that also suggested similar claims with other large data sets. So, could we find the calm after that storm? And I think ABCD gave us a really good opportunity to use subsamples of the data to really address, like, do you really need thousands of individuals? What does this mean for other data sets and even future research endeavors? Do we always need to be carrying out ABCD-like studies, or can we use studies like ABCD to help inform smaller studies? What is the sample size required? So I'm going to give you one example from work that we actually published in our group with myself, Andres Dale, and Terry Jernigan, and I'll highlight that paper in a couple of slides. But just some considerations, just so you can kind of keep thinking about these things. So I've talked already a few times about univariate versus multivariate methods and how we can maybe boost these effect sizes so you don't need thousands of individuals. When we're thinking about brain imaging data, we need to also consider the resolution that we're using. So a lot of people have turned to region of interest based analyses, but we actually have even higher resolution or kind of more fine resolution of our imaging data in ABCD that we'll talk about. There's many different imaging modalities that we collect. And then again, not just on the imaging side, but what are we actually looking at in terms of behavior? So bringing it back to that first point, univariate versus multivariate methods, what is multivariate? So univariate approaches, you can kind of just think of this as looking at a single association at a time. So I've highlighted just one region of interest here within the precentral gyrus, kind of the motor strip along the cortex, and just linking that to maybe some fine motor function or fine motor behavior. So this is really looking at one association at a time. This could be between individual regions of interest or individual voxels within the brain or vertices, points along the brain. But we know that behavior is complex, and it isn't the case that behavior is just localized to one brain region. And brain regions also do not act in isolation. There is a very high covariance structure, regardless of what imaging modality you're looking at. Regions are highly connected to each other. So really, our brain is not these regions aren't acting in isolation. So multivariate approaches are kind of approximating more what the brain is really doing and trying to gather that signal across the entire brain and really takes into consideration that high covariance structure within the brain. Whether you're looking at just brain structure, there's a lot of regions that are highly correlated with each other in terms of their size or surface area, cortical thickness. But we also have, if you're looking at like functional MRI, a lot of regions tend to co-activate with each other, right? So that's what I mean by this covariance structure. So multivariate approaches will identify the major sources of variance in your data, and it will describe major effects of interest in the data in terms of these sources of variance. So another way of saying this is you're really putting in patterns. You were not just putting in one single metric, which is where the multi obviously comes from. I also wanted to mention the spatial resolution of imaging data that is available in ABCD. I think a lot of publications are using the tabulated region of interest-based data, which is very understandable, especially for those that might not be coming from a neuroimaging background. And also, up until release 6, the tabulated imaging data was probably the most accessible, and that was actually released and sort of easy to use for folks. So this is going to include across all of our modalities, which include structural MRI or SMRI, diffusion MRI, resting state functional MRI, and task functional MRI, where we actually have within CSV files or text files that you actually get different brain-derived or MRI-derived features like cortical thickness, sulcal depth, activation within particular brain parcels. And then we have many atlases that are available in ABCD. All of this stuff is in the documentation, so I'm not going to go over all that, but tabulated data is what a lot of people have used. We do have more fine-grained resolution that we can look at and that are not going to confine our analyses to these boundaries, which can actually have a real impact on your discovery of associations with behavior. So we have vertex-wise data across the cortex. So you can see that in the kind of second schematic along the left. You can actually get this data for structural MRI, so you can get across thousands and thousands of vertices or points along the brain, things like thickness, area, sulcal depth, contrast between gray and white matter. You can get also this data for diffusion MRI, kind of pericortical within the cortex and within superficial white matter, tissue organization or tissue microstructure across the cortex. And you can also get this vertex-wise data for task functional MRI. So for instance, beta weights for each task-based contrast, for instance, 2 versus 0 back with the and back working memory task. And then we can go even more fine-grained resolution with voxel-wise data. So now we're kind of looking more in 3D, and you can actually look within subcortical structures. For instance, with structural MRI, we're now releasing this voxel-wise data for morphometry with the Jacobian determinant, which you can kind of just think of it as volume. So how much you have to shrink or expand a particular structure to fit an atlas. And then we also get this voxel-wise whole brain data for our diffusion MRI metrics through diffusion tensor imaging and the restriction spectrum imaging model. Again, go read the documentation for all those, but just so you're aware. And then we also have our correlation matrices for functional MRI, where you're looking at correlations between different parcels in the brain. So again, you're working with like hundreds, thousands of connectivity measures or correlation measures with our time series data. So you've got all sorts of parcellations there. So your tabulated vertex-wise and voxel-wise data, you're essentially getting, all of your observations, participants, and sessions across, however many regions or vertices or voxels you're looking at. And then the correlation matrices, you actually have, it's more of a three-dimensional structure where you've got your region by your observations. So lots to consider there with imaging data. So going back to the example I mentioned that I would highlight to really boost these our predictive power and what sample sizes do we really need to predict behaviors of interest in ABCD. So this is a paper that we published with Tim Brown, Terry Jernigan, and Anders Dale at UCSD last year. And using more of this high-dimensional cortical data and also multivariate methods and all these considerations that we've been talking about so far. So let's see it in practice. So what we wanted to do here was understand, which imaging measure is most predictive of general cognition, which we'll go over in a moment, and also see if we could replicate this idea that you need thousands of subjects to reliably or reproducibly measure things like cognition, or if we could do better with a smaller sample. So for our imaging measures, we included across structural diffusion MRI, and functional MRI. So we have things like surface area, cortical thickness. We have some measures of restricted diffusion, both within the cortex and in superficial white matter. And then we also have data from functional MRI in this paper where we're looking at 2 versus 0 back beta activations. And then we also have data from resting state fMRI, where depending on the participant, we get up to 20 minutes of data where individuals are staring at a crosshair. And then I'll just mention quickly, if you're not as familiar with the task fMRI data, for the working memory task 2 versus 0 back, you are asked for the 0 back condition if every image that you're seeing is the same as a target that you were exposed to at the very beginning of the trial. So it's very easy because you just have to keep in mind that target all the time. Versus the 2 back, you have to see whether the stimulus you're looking at is the same as the stimulus that was 2 trials ago. So you really have to keep a little bit more in your working memory. And then there is an emotional component to it because there's positive versus negative versus neutral faces versus places. For our outcome, we were predicting a total composite score from the NIH toolbox. The reason being this kind of captures a general cognitive ability that makes it a little easier to compare against different studies. And this is also what was predicted or associated with brain in the Nature paper from Marek and Turvo-Clemens et al. And just as a refresher in ABCD, our NIH toolbox total composite score is really a composite over 7 different subdomains or kind of tasks within the toolbox. And then those can kind of be collapsed a little bit more into crystallized versus fluid composite scores. So we're really just taking this total composite score here. For our framework, we have our imaging measures that I showed you a couple slides ago, predicting our total composite score. And we are looking both at univariate and multivariate methods here. So you can see the sample sizes that we have per imaging modality in the top left. Our sample sizes do differ depending on the quality control that is very imaging modality specific. So you do end up losing more folks for the functional MRI, but we used as many people as we could from the baseline visit in ABCD. So first, what we did is just estimated the univariate associations across each vertex in the brain, whether we were looking at cortical thickness or restricted diffusion or task fMRI. And then for resting state, we actually put in the correlation matrices. So we estimated those univariate betas between our brain and behavior or cognition. For multivariate prediction, we used least squares and ridge regression by using a common function in MATLAB called fit-r-linear. And we did this with 100 random splits of 90% of our sample into train and then 10% into our test. And really important, and I'll mention this again, we really wanted to use as much of the data as possible. So we did not run this on singletons, but we did take into consideration the fact that we have this complex family structure in ABCD. So if we had members of the same family or that had the same family ID, we kept them together within training or kept them together within tests. So we were never training on one family member and then testing it on, a family member. So we were really keeping families together in that way. And then our metric for prediction performance was the correlation between the observed and predicted behavioral score in our replication sample. So that 10% replication or test set. Okay, so this is a summary of the results that is published in that paper. And on the left in panel A, you have our multivariate out of sample prediction, where you have your R value or prediction value along the y-axis, and then along the x-axis, you have all of our imaging modalities, and they're colored. Structural MRI is grouped together in blue. So we have surface area and cortical thickness. Our diffusion MRI, both within kind of cortical versus within superficial white matter, RND and RNI respectively are in green, and then in purple you have our functional MRI. You've got your N-back or emotional N-back in resting state, and then univariate associations in the right panel. What you can see right away is that you have pretty high predictive performance, the highest in the emotional N-back task. And you do get pretty high performance with resting state, but you can notice that, or hopefully appreciate that the standard deviation bars are quite variable across our 100 iterations. The numbers that are on top of each of these bars are the sample sizes required to detect these effects with 80% power. So 80% power is sort of the standard that typically power calculation analyses are kind of asked to do. So 80% is typically what we aim to strive for. So with our task fMRI, emotional NBAC task, we're able to predict total cognitive score with just 41 subjects in our replication sample. So in our test set, we're able to detect this effect with 80% power with just 41 subjects. Versus if you look univariate, so you're not putting in that entire pattern of associations into the predictive algorithm, but you're just looking at sort of one association at a time, you need over 100 subjects, which isn't too bad with functional MRI for the in your replication sample. But you can appreciate if you look at, for instance, the diffusion MRI measures, you do need upwards of hundreds, even maybe thousands of individuals to assess these effects with general cognition. So this is sort of where that thousands of individuals are required if you are using potentially univariate methods. So you really, really can boost your effect sizes, and you do not require as many subjects in your test set with multivariate methods. So how am I gathering these sample sizes? So this is just coming from power curves that you can generate with like any statistical package that you're using. There's also a lot of online packages that just allow you to put in, what effects that you're getting, and what power level that you are, striving for, and then it'll kind of output the sample size. But just as an example, again, bringing it back to the emotional NBAC test, since that was our sort of best imaging modality predictor, here you can see that if you want 80% power or 0.8, you would need 41 subjects along that x-axis there to achieve that effect. With total cognition in our sample. Whereas for univariate effects, you can see everything is shifted over the right. You need way more subjects. So why is multivariate doing better? And I think it's telling to always look at your data, right? But it's telling to look at your univariate associations. And so this is across the brain, all of these effects across the brain by imaging modality with general cognition. And you can see, for instance, NBAC is a really nice example here. You see, some patterns of increased, for instance, like dorsolateral medial prefrontal and dorsolateral prefrontal precuneus are associated with better cognitive performance, higher activation in those regions associated with better cognitive performance. And then you see some blue negative associations, but it's not just, one brain region here that is associated with general cognition. It's really this pattern of effects. And with multivariate prediction, you can actually put in this, entire univariate pattern. And then you can see really, really tiny effects, for instance, with cortical thickness and our restricted diffusion measures in our middle columns there. So those are boosted with when you actually just put in that entire pattern rather than looking at individual small effects on their own. Okay, so everything I have said so far was really pointing to the sample size that you require in your test set. So you still, based on what I've showed you so far, you still need thousands of individuals in your training set with ABCD to get those effect sizes. So what if you only had 100 subjects in your discovery sample, right? Do you actually need thousands of individuals even in discovery? So here what we did is we looked at our predictive performance as a function of different sample sizes in our discovery set, and then again tested the predictive performance in our replication or test set. And again, just highlighting the emotional NBAC as an example, with 100 subjects in your discovery or train set, you can actually predict cognition with an R of 0.34, which equates to having about 66 subjects in your test set. So now at no point do you need, either in train or test, thousands of individuals. You can get by with 100 subjects and 66 subjects in your replication. Whereas if you look at univariate, which is the dotted line here, I was describing results for the solid line a moment ago. But with the dotted line, you can see that with univariate, you need a little bit more time to catch up. So with 100 subjects in your trained set, you still would need 400 subjects in your test set to detect that effect, just because, again, we're working with much smaller effect sizes when you're doing just one region or one vertex at a time. So you're really getting this boost with multivariate effects once again. So the good news is that reproducible brain behavior associations can indeed be measured with multivariate methods, not only in large data sets like ABCD, but in smaller replication samples, which is really, I think, reassuring for many people that are addressing, these brain behavior associations in much smaller samples in their own labs, and then also just the wealth of data that we have from other data sets. We don't need to discard them. They're still very, very meaningful. So I won't reiterate this because I just spoke about it. But I was only showing you a prediction of general cognition, which is a measure that an outcome that a lot of people have looked at. But if you look at, some other measures, for instance, that are tapping more into mental health concerns at baseline, you still do potentially need thousands of individuals. So there are methodological considerations that we're going to talk about to boost these sorts of effect sizes. So we're looking at sample sizes here required in your test set. So the bars are kind of smaller for multivariate and cognition, which are our first four variables. But then if you look at like child behavior checklist, externalizing and internalizing symptoms, and a general sort of P factor of mental health concerns, you do need thousands of individuals with ABCD. And this also, keep in mind, is baseline. So a lot of youth were not showcasing a lot of mental health concerns yet, or there wasn't maybe as much variability to play with. And we are also just inputting every single feature within the brain. So there's ways to kind of fine tune these models to be a little bit more neurobiologically relevant for the outcomes of interest that we'll talk about soon. So one of our objectives also for today was to understand the contribution or impact of different sociodemographic covariates on these predictive models. So in the cerebral cortex paper results that I was showing you, we were just covariating for age, sex, and scanner. So we kind of call that our base covariates, the things we sort of need to covary for. If you're not looking at sex stratified, obviously your age associations. But obviously, a lot of people have been considering, what are the effects of things like socioeconomic resources, things like race, ethnicity. For imaging data, there's, you always need to think carefully about whether you do need to be, or why you're covariating for variables like race and ethnicity, which I believe are covered in other ReproNim lectures. So this example that I'm showing you from Vicky Zhao, who was a PhD student in our lab, she was predicting general cognition, so same measure, but she was using, again, resting state functional connectivity, which I'm highlighting in the left here. And then she was looking at beta activation contrast across three different tasks. So NBAC, stop signal task, and the monetary incentive delay task. So just focus your attention on sort of the outer red squares right now. For our predictive model of total composite cognition, we get an R squared of about 13.1% with resting state functional connectivity, and we get even higher predictive power or accuracy with NBAC in the MID task. So you can see the numbers, 18.2% and 15.7% respectively. So that's the gray bar. That's when we have, everything in the model. Age, sex, we have top 10 genetic principal components that tend to be associated with brain structure and possibly with these functional MRI measures also, and then our socioeconomic resources. Now, the unique contribution of our imaging measures are in blue. So you can see that a lot of the variance explained of, imaging measures explaining total cognition, a lot of that variance is being actually explained by our sociodemographic covariates, and you really get this drop down to the blue bar when you're only looking at sort of the unique contributions of imaging measures. Here's another paper from our group. It was published a few years ago, but this is sort of just reiterating really that message, but with structural MRI rather than functional MRI. I know that the models are very small here, but hopefully you can, zoom in on the slides and look at the paper as you wish. But here, Claire Palmer, who was a postdoc in our group several years ago, was looking at the, impact of these different sociodemographic covariates on imaging prediction of again, cognitive scores. Here she actually stratified by fluid versus crystallized composite scores, left and right respectively. And then the A row is for cortical thickness, and B is when you have surface area as your predictor. So the first model, the darkest bar, is sort of when you put everything in there. So this includes age, sex, scanner, parental education, income. She had race, ethnicity as variables, as covariates rather than the top 10 genetic principal components. And then if you put in the actual imaging measures also. So the black bar is sort of the total, percentage R squared that we get of variance explained in general cognition. And then the next two models, the sort of light, still deep gray bars, are just the predictive kind of accuracy of if you just have behavioral or just sociodemographic variables in there. So you see this drop, but obviously, and then if you just have, your imaging measures, you see that there's really only a tiny amount of variance that is explained that's like completely independent of these sociodemographic factors. And you can see in the blue sort of highlighted message that she had, where controlling for sociodemographic factors reduced the variance explained in the composite scores, the cognitive composite scores, by 4 to 5-fold for cortical surface area and by 4 to 8-fold for cortical thickness. So your sociodemographic variables and how you're actually, or which covariates you choose to input may have a very large impact on your interpretation and predictive power. And there is this very, strong interplay between brain and a lot of these sociodemographic variables when you're trying to explain something like cognition. So these are really important things to keep in mind. Um, Considerations, of course, methodologically speaking, you need to think about what predictive algorithm you're using. It is way beyond the scope of this lecture to go through machine learning and all the sorts of models that exist. But I would highly recommend you check out Dr. Gael Vadakwo's lecture on the fundamentals of machine learning. That is part of the ReproNim kind of repository of lectures. And I just want to reiterate one more point again, because repetition can be key. If you are using ABCD data and you're including everyone, do not forget that we have repeated time points, right? And we also have these family IDs. So there's siblings, there's twins, even triplets. So really ensure that you are never, training your data on one family member and that family member is also in test. You really want to keep, everyone sort of together. I do want to touch on imbalance data and subsampling strategies. This isn't particularly important for if you are working with maybe a binary outcome. So this is something that is close to home for me because I work in psychiatry and I'm really interested in eating disorders and psychotic disorders also. And if we are, looking at categorical variables, you might have a really big imbalance in your outcomes. So you might have, only a few hundred subjects that are meeting criteria for a diagnosis or for some mental health concerns. And then you have a lot of other individuals within ABCD that don't endorse, those symptoms. So, and that also means that you might have very large imbalances in your demographic covariates. So if you have, on top of having let's say, a high level of ADHD symptoms in one group and low in the other. And then they also tend to have significant group differences in demographic variables. If you are trying to, achieve high predictive accuracy with imaging data, you want to ensure that you have not only just balance in terms of your sample sizes, but you're not just predicting your outcome unintentionally based on sociodemographic differences. So there might be ways of, for instance, oversampling or undersampling your majority class or oversampling your minority class in ways that are trying to also balance out some of these sociodemographic imbalances, if that is the case. So Ariana Gard had a really elegant, wonderful paper of considerations around sampling based on sociodemographic variables. Here I'm highlighting in your actual like data when you have a class imbalance, how you can over versus undersample. There's also ways that you can kind of synthetically add data in oversampling through a method called SMOTE. And you can also take into consideration this imbalance, if you have class imbalance in your outcome, through your actual machine learning algorithm itself. So there's cost-sensitive machine learning models as an example. Just something to keep in mind because you do have such a wealth of data to choose from ABCD, but it's still possible, depending on your outcome, that you might have these imbalances in your outcome. Okay, so we've talked a lot about, the power and potential benefit of using multivariate methods, but I am not suggesting that that should be always your go-to. You really do need to ask yourself, what are you trying to achieve with your question, with your research question? So when do you use multivariate methods versus kind of more simpler or traditional null hypothesis statistical testing? And I think that answer lies in kind of a trade-off of what you're looking for. This was really nicely summarized by a paper that was published a few years ago in Molecular Psychiatry by Tea Vibulia and colleagues, where they kind of elegantly demonstrated that there is this trade-off between prediction accuracy and interpretability. So predictive models may be really useful if you are looking for like early risk markers, or just trying to detect which individual might be, either at higher risk or for a condition based on sort of earlier data, imaging data, for instance. But just a predictive model on its own won't necessarily point to biological mechanisms, although, you can get into fancier algorithms that actually do weight or show you feature importance in your models. So there might be some cases, if you do really want to understand mechanisms, or you really want to understand the neurobiology, that it might be better to just stick to more traditional null hypothesis statistical testing and assess the interplay between different factors that could be informed by predictive models for the behavior or outcome you're trying to understand. So I've shown a lot of kind of data-driven methods, especially the multivariate prediction methods that we had in our cerebral cortex paper, but theory-driven approaches are really valuable, and I think there's a way that we can merge both. I would really encourage, if you are using a theory-driven approach with ABCD, really to provide appropriate justification and evidence, consider pre-registering your hypotheses as well. So for the next, kind of the last section of this lecture, I'm really going to talk about using ABCD alongside other data sets. Before I get there, though, we're going to talk about brain state. So one more consideration before we integrate other samples. Brain state. So this is really interesting. There was a recent paper by Grosshauer and colleagues in molecular psychiatry last year that showed that actual therapeutic effects of, in this case, it was transcranial magnetic stimulation, were really dependent on what the participant was actually doing. So I'm going to get to the application or so the interpretation of this for ABCD. But here, if you haven't heard of transcranial magnetic stimulation, it uses magnetic fields to stimulate brain regions and neurons within the brain in a non-invasive way. So you really are just like putting this device over the person's scalp. There's no surgery, there's no cutting required. Transcranial magnetic stimulation is kind of a promising approach for a lot of psychiatric and neurological disorders, for instance, depression. And people have used transcranial magnetic stimulation to impact or try and stimulate the anterior subgenual cingulate, which is very, very, very deep in the brain. So in the end, you're actually stimulating more the kind of lateral prefrontal cortex. But what they found with both transcranial magnetic stimulation and they also had a functional MRI paradigm where if the individual was just resting or just doing a simple zero back trial test, the transcranial magnetic stimulation was not as effective compared to when a participant was actually engaging in a more challenging working memory task, the two back. During TMS. So there was actually increased modulation of clinically relevant regions within the brain and that are relevant for depression when a participant was engaging in that more challenging task. And kind of bringing it back to, the functional MRI paradigms that we have in ABCD, you can choose obviously from resting state functional MRI, but we do have a lot of task-based functional MRI data also. And thinking about if you are using these brain imaging measures to predict or just associate with a behavioral variable of interest, depending on the person's brain state, you actually might get different, predictive power. So if someone is just resting in the scanner and staring at a crosshair the way that they do with resting state fMRI, perhaps it might not be as predictive with general cognition, which is what I've shown you, and in the references that you'll get along with these lecture slides, many papers have shown that you actually do get higher prediction with task-based functional MRI because you are engaging the brain in this challenging task in a more kind of mindful way, and that might be more associated with cognition. There's also resting state functional MRI paradigms that have movie watching in it. We don't quite have that in ABCD, but it is a consideration also when analyzing existing data sets. Okay, so now we're going to get to actually incorporating other samples. So what I've already kind of shown you a glimpse of is sort of direction one, using a large sample to inform prediction in a smaller sample, but I'm also going to flip that framework around and showcase how we could use smaller samples to inform where we should look in ABCD also. So going through the first, there's several examples that I'll kind of go through here. There's many papers and references that you can look at in more depth. So I'm just kind of going to give you an overview. But as one example, you could use, ABCD to look at these effects across the entire brain and kind of port those weights over and create a summary score in smaller samples. That's very akin to what folks do in the genetics world. So in genetics, we have these, mass univariate studies called genome-wide association studies that are associating, every single location across millions of locations across the genome and associating their genotype across thousands of individuals to a particular behavior. And then you can actually take all of those effect sizes across the tiny, tiny effect sizes across the entire genome and then summarize them into like one polygenic score, if you've heard of polygenic score. So you can do something very similar with the brain, even with ABCD, and you can aggregate these tiny individual brain behavior effects and then use those weights from your large discovery sample to derive something like a poly brain score. So we have some papers in the group that have called this the polyvertex score because as the name suggests, it was derived on cortical vertex-wise data. There's a paper from Nora Byington et al. That called this the polyneuro score. They're all very similar flavors, right? So you can either in a univariate way just summarize all of those mass univariate effects across the brain, or there's also different sort of more nuanced ways of doing this with multivariate approaches again, where you actually account for the inherent covariant structure across the brain. So really taking into consideration the non-sparseness of your brain signal on behavior. So this was done with a Bayesian polyvertex score by Vicky Zhao, again, that PhD student I mentioned earlier. Okay, so you can also, and we're seeing this more now, and I think this is really, really important, particularly for like psychiatric imaging, which is something I'm very interested in. And I think a lot of people using ABCD are interested in mental health too. So using these large data sets to inform mental health prediction in smaller samples. Maybe you'll recall that I showed you in our cerebral cortex paper that we really didn't see very high predictive power of, for instance, CBCL internalizing and externalizing symptoms, but that just may be because baseline ABCD data is not the right target for internalizing, externalizing symptoms with the imaging data we have. So this is a really elegant study by Siddhant Chopra that was published last year in Science Advances. I've also included the link to the GitHub that has the actual code that is underlying this paper. And what Siddhant did was he used resting state functional MRI data in 37,000 UK Biobank participants. So this is not ABCD, but you can absolutely generalize this framework to ABCD because we do have resting state fMRI data. So using that resting state fMRI data, he trained a deep neural network. Could be another machine learning model, but he trained a machine learning model, or sorry, a neural network here. Could be another predictive model. And then he used that model and ported it over to 3 psychiatric cohorts that were much smaller, obviously, than the 37,000 individuals. And I also mentioned that the deep neural network was really trained on dozens of different phenotypes across sort of psychological constructs, cognition, etc. Whereas the target outcome in the psychiatric cohorts was a general cognitive score. So they weren't even the same necessarily outcome, but the idea being within this meta matching framework, as they call it, using a very, very large sample with many different phenotypes that are collected to kind of try and rather than put in the entire brain signal, try and see in a data-driven way which brain regions have more specificity and are actually helping power the prediction in UK Biobank. And then now you have a more biologically informed model when you're testing it in your kind of independent samples. So they had 3 psychiatric cohorts here, and they actually saw much higher predictive performance compared to chance in all 3 of those samples. And then even some, methodological considerations there, but that's basically what panel B in the middle is showing you. In those 3 psychiatric data sets, you get pretty good predictive performance. And this is all informed by that deep neural network that was trained in UK Biobank. And then in B, you really have the same sample. So everyone from the Human Connectome Project in train and test, and everyone from the Transdiagnostic Connectome Project in train and test in the middle. And then we have another sample consortium from Neuropsychiatric Phenomics. But if you actually kind of do some crosstalk and have, you train on HCP, but then you actually test it in, CNP, that 3rd psychiatric cohort, you actually still get pretty good generalization to these independent psychiatric cohorts, even when you change which cohort is in train and test, all again informed by this large data set. So very cool, and it's very promising that we're getting, better generalizable performance in totally independent test sets, which has been really a challenge for the field. Here's another example of using ABCD study data to help benchmark smaller independent samples. This was really a very high impact paper that used ABCD data amongst many, many other scans from Richard Bethlehem and Aaron Alexander Block's group. And they actually used over 100,000 individuals and over 123,000 MRI scans, basically spanning birth to 100 years of age, included ABCD data. But the beauty of having these large data sets is like you can actually derive trajectories across age with, for instance, cortical thickness. We've been talking a lot. This paper used volumes, but they also did look at cortical thickness, volume, surface area, all these things. So you can really think of this as very similar to growth charts, for children. Like, are children developing at a normal rate based on their height and weight for their age and sex? So you can use these large data sets to, model what a typical, trajectory would look like and sort of the deviations around that. And then they have a tool on brainchart.io that's part of this paper, where you can actually incorporate your own sample and you can estimate these study specific offsets and see how much does your study and individual participants actually deviate from this trajectory that was estimated through this large sample. And then now there's even more sort of developments happening through something called the reproducible brain charts, that was just posted as a preprint in bioRxiv this year. So, it's not clinically actionable yet or anything like that, but you can see again how these large data sets are really, really coming together, even in tandem with other large data sets to help inform and maybe benchmark smaller studies. So I'm going to flip this framework around for a moment, and showcase a really cool example of how we can use existing or new developments in smaller samples to actually inform where we need to look in something like ABCD. So this is an example coming from precision functional mapping, which really prioritizes quantity of data per individual. So rather than just getting, one scan or like 20 minutes of functional MRI data, you get hours of data across, many, many sessions per individual. So this is really resting state functional MRI data. But what you're seeing in this figure to the left is kind of an average of, known kind of canonical networks or functional brain networks, and their anatomical sort of location. So that's an average that a lot of people have sort of cited. But if you actually look at individual brains, so now you have 8 individual brains here, you can see that the, anatomical specificity of like where all of these brain networks do vary by individuals. So there's some individuals that kind of have like, more kind of larger default mode networks, or they have, sort of more territory that is associated with that network and then other networks shrink. So there's really this individual specificity. There's some arrows on this plot that are kind of pointing to these individual differences. So you really get much more precision in these anatomical maps based on these functional MRI long sequences and acquisitions. So this is a paper from Lynch and colleagues that was published in Nature last year, and they use precision functional mapping to identify a highly replicable finding of an expanded frontostriatal, or sometimes it's called salience network in depression. So what they did is, if you look at panel A in the left, that's sort of your what the salience network, where it typically lies when you're looking at kind of a group average and healthy controls, and it's made up of like the anterior cingulate cortex and lateral prefrontal cortex and anterior insula. And then if you look at individual, data sets from folks that underwent approximately 10 hours of functional MRI across 22 sessions, give or take, depending on the person. But if you look at individual maps, and you actually map that frontostriatal network, it tends, or it seemed to be, with this precision functional mapping in individuals with depression, it seemed to be expanded. And you can see that, just eyeballing at it, that the frontostriatal salience network is bigger, but the authors actually went ahead and quantified it. It does take up more sort of territory of the cortex when you're looking at these individual precision functional mapping data sets. And then they also replicated in 3 different data sets. And you can see this on the right-hand panel here, with major depressive disorder, MDD data sets with kind of higher, or this expanded frontostriatal network compared to healthy controls in terms of the percentage of cortex it took up. And if so, that was in more of these precision data sets. But if you actually take data sets like ABCD, this slide doesn't yet include ABCD, but data sets that have a similar protocol in terms of their functional MRI data, they actually were able to find this expanded frontostriatal salience network in if you were just looking at conventional acquisitions of functional MRI. So this is where it gets really cool because they did use ABCD data here. And uh, based on this small precision functional mapping data set and this, really this finding of this expanded frontostriatal network, the authors then asked, well, can we already see these differences in the frontostriatal network in ABCD prior to onset of any depressive symptoms, which we can actually answer in ABCD. So what they did is they took, so it's a very small sample from ABCD, but they took 57 individuals that had clinically relevant depressive symptoms as measured by the Child Behavior Checklist by year 3, but had absolutely no symptoms at baseline or year 2. So this is really like a true or capturing onset of depressive symptoms. And then they had a randomly selected group of healthy controls, also 57 subjects that had depressive scores well below the clinical cutoff. So you can see this in the left-hand kind of schematic. Here's in red is our MDD group from ABCD, and in blue is those that just have consistently low depressive symptoms. On the right, you can see the size of the frontostriatal network, again, really informed by that precision functional mapping data set. And you see in red, those that have future at year 3, those that have clinically kind of relevant depressive symptoms already at baseline and their year 2 follow-up in their imaging data, you see that expanded or higher kind of take up of the frontostriatal network versus those that never have depressive symptoms. They have a smaller kind of what we would expect the frontostriatal network to look like. There's interestingly no difference in that sort of MDD group between the 2 time points, suggesting that this increased frontostriatal expansion is really potentially a trait or something that's kind of a risk factor preceding onset of depressive symptoms. So really cool way of flipping that framework of going from small data set to large. So some take-home messages as we wrap up. Large samples are incredibly hard to collect, so we're super lucky to be able to work with ABCD data and how you leverage large existing data sets to help inform other independent studies is really dependent on context. We've talked a lot about, using large data sets like ABCD to discover brain patterns, apply weights to another sample, drive trajectories in ABCD to benchmark independent samples against, and then flipping that framework around as we just discussed. And I would also just acknowledge that rather than focusing just on increasing sample size, it's also important to improve the quality of our chosen neuroimaging and clinical measures to boost effect sizes. So this is where I was talking about, what state is the participant in when they're engaging in a task, for instance? That's going to be really important. What behavior are we looking at? Are we looking at a mental health concern way before, that doesn't have a lot of variability yet in our data set? Are we, leveraging information on cognition to then predict a similar score in another data set? And then, of course, there's a lot of decision points along the way in terms of your analysis. So I will just point you, or I'll leave you with this paper that I published last year. That's a review paper in neuropsychopharmacology that actually has a flow chart that I show here of some decision points that you can think of to improve or to boost your effect sizes. Again, the data is the data. So if there is no effect there, if it's very small, there's not much you can do, but there are, some considerations and steps that you can take to potentially boost those effect sizes with multivariate analyses, choice of imaging paradigm, resolution, behavior, everything that we talked about today. So revisiting those objectives, hopefully now by the end of this hour, you're able to assess and interpret effect sizes from brain behavior studies. You are a little bit more familiar in differentiating between univariate and multivariate methods. You're able to critically assess the impact of different demographic covariates and subsampling strategies on brain behavior prediction results. And also, hopefully this has inspired some research study designs that you can carry out yourself on leveraging ABCD data with other data sets. And with that, I'd just like to say thank you so much for tuning in. I hope you enjoyed this lecture. And if you have any questions, please do not hesitate to contact me. My email is up there, and I am also on Blue Sky, so feel free to give me a follow. Thank you so much.